It is my distinct honor to introduce the keynote speaker of today's symposium, Dr. Lewis Cantley. And we are delighted to have him and deeply appreciate his participation in the symposium. Although he scarcely needs introduction to this group, I would like to welcome him to this symposium entitled From Pathways to Therapies, and this is very apropos since his groundbreaking research that most of you are familiar with on PI3 kinase pathway, which directly resulted in improved therapies for a variety of diseases, including cancer. So Dr. Cantley is Mayor Director of the Sandra and Edward Mayer Cancer Center, Weill Cornell Medical College, and the Ronald P. Stanton Clinical Cancer Program at New York Presbyterian, as well as Margaret and Herman Sokol Professor of Oncology Research and Professor of Cancer Biology Medicine. That's a mouthful. So Dr. Cantley is no stranger to the Boston area. He graduated summa cum laude BS in chemistry from West Virginia Wesleyan College and obtained PhD in biophysical chemistry from Cornell University. He did his postdoctoral research at Harvard University, and then he was appointed assistant professor in biochemistry and molecular biology in 1978. He became a professor of physiology at Tufts University in 1985. Then he returned to Harvard Medical School as professor of cell biology in 1992. He became chief of Harvard's new division of signal transduction, and he was a founding member of the Department of Systems Biology in 2002. In 2007, he was appointed director of the Beth Israel Deaconess Cancer Center, and he joined the faculty of Weill Cornell Medical College and New York Presbyterian Hospital in 2012. So we are sorry he left the Boston area, but I am told that he comes here very frequently. So Dr. Cantley has made significant advances in cancer research stemming from his discovery of the signaling pathway phosphoinositide 3 kinase, PI3 kinase, in 1984. His pioneering research has resulted in revolutionary treatments for cancer, diabetes, and autoimmune diseases. He's an author of 400 original papers and more than 50 book chapters and review articles, and he's a fellow of the American Academy of Arts and Sciences and a member of National Academy of Sciences. So with this short introduction, please welcome Dr. Cantley to the symposium.
Thank you. It's a pleasure to be here and get a chance to talk about what we're doing with PI3 kinase and new approaches to understand how to use inhibitors of PI3 kinase for treating cancers. I do have a disclosure. I will be talking about some drugs being developed by Novartis as PI3 kinase inhibitors that we're using in clinical trials. And I want to start by acknowledging my collaborators that contributed to the work that I'll be talking about today. The part on PI3 kinase and PARP inhibitor drug combinations has been a collaboration with Gerber-Wolf's laboratory, in particular work by Hashish Juvekar there. I'll talk quite a bit about our Stand Up to Cancer funded clinical trials, particularly collaboration with Ursula Matsoulianis at Dana-Farber. Gerber-Wolf is also involved in these clinical trials as well, and Eric Weiner in particular as a leader in these trials, and also a collaboration with Ingrid Meyer and Carlos Artiega that's led into a neoadjuvant trial in breast cancer. So the talk has two parts to it. The first part will be a background on PI3 kinase and the pathway that it's in, and then the second part will be the clinical trials that have been initiated through our Stand Up to Cancer team. So just to remind you what PI3 kinase is, it's a lipid kinase, and it phosphorylates the 3' position of the inositol ring to generate this triply phosphorylated lipid. We actually found the activity at co-purifying with a variety of oncoproteins, including VSARC, polyamidyl T, PDGF receptor, et cetera, back in the mid-'80s. But it wasn't until 1988 that we realized it was carrying out a reaction that had not previously been known to exist. Prior to our discovery, phosphatidylinositol was known to be phosphorylated to 4 position and the 5 position. And in fact, that was discovered in 1949. But it was not known to be phosphorylated at all at the 3 position. So what we discovered was that not only can PI3 kinase phosphorylate to 3 position, it can do it if the 4 or 5 positions are already phosphorylated. So it can make multiple forms of phosphoninositides that weren't previously known to exist. But the one that has been of most interest is this triply phosphorylated lipid, PI345P3. About 10 years after we discovered PI3 kinase, the tumor suppressor gene P10, which had been identified in the mid-90s as a tumor suppressor, and the sequence predicted it was a phosphatase, but it was thought to be a phosphotyrosine phosphatase, and does have some phosphotyrosine phosphatase activity. But it turns out that the favored substrate by orders of magnitude is PIP3, and this was work done by Jack Dixon's laboratory. Jack's really a brilliant enzymologist and had worked on phosphatases for much of his career. And he realized that this was a very poor protein phosphatase and began looking for other substrates and discovered it could dephosphorylate the 3 position of PIP3. And so that combination of either activation of PI3 kinase or loss of function of P10 results ultimately in the same thing, an elevation in this triply phosphorylated lipid. So what does this lipid do, and what does PI3 kinase do? And evolution is, by following the evolution of this enzyme, it's told us a lot. So there is an enzyme in yeast that will form PI3P, but there is no enzyme to make PIP3. So PIP3 is not produced in Saccharomyces cerevisiae. It's not necessary for the normal functions of a eukaryotic cell. If you ask where does it appears in multicellular organisms. And if you ask what's in the pathway in flies and worms, it's in the pathway for insulin, IGF-1 signaling. So this pathway evolved for the purpose of allowing a multicellular organism to decide what cell should take up the nutrient, particularly glucose, that is needed for that cell to grow. For yeast, it doesn't matter because yeast grows whenever it has enough nutrient. But for a multicellular organism, you have to make a decision. When does a cell take up nutrient and when does it not? Because that will determine when it can grow. And so it's insulin IGF-1 signaling really that evolved to control this pathway. Now in humans and mice and more advanced organisms, there's more than just insulin that can activate this pathway. Many growth factors can do it, PDGF, EGF, et cetera. But still, insulin is the champion, and insulin and IGF-1 are dramatic activators of PI3 kinase compared to any other stimulus. So the purpose then is to produce PIP3, which ultimately turns on glucose uptake and also controls glucose metabolism. And this will lead to cell growth and cell survival. And as long as P10 is around, the cell will only grow when it's told to grow. P10 will shut the system off after a certain amount of signaling has transpired. Now, it turns out that if you cannot activate PI3 kinase, it results in insulin resistance and ultimately can go on to form type 2 diabetes. This, in fact, is the most dramatic disease in the world now. It's growing at a rate that's really frightening as people are overeating, and particularly eating sugars that tend to suppress insulin signaling through negative feedback loops. And this invariably is a consequence, this insulin resistance, is a consequence of negative feedback shutting off insulin signaling so that you ultimately cannot reactivate PI3 kinase and take up glucose in the muscle, and you can't suppress gluconeogenesis in the liver. But here we're talking today not about insulin resistance, but rather about cancers. And in case of cancers, it's not a problem of failing to activate PI3 kinase. It's quite the opposite. It's hyperactivation of PI3 kinase. And this can happen in a variety of ways. It can happen because the growth factors are overexpressed, or because the receptors are mutated or overexpressed, or PI3 kinase itself is mutated or overexpressed, or P10 is lost. All of these will result in high levels of PIP3, which will then tell the cell to take up nutrients and to grow. Now, what does this lipid do? And as I said, we discovered the lipid in '88, but we didn't know what it did. It wasn't until about 6 or 7 years later that we began to get an inkling of what it was doing. And our first real insight came from the protein kinase, BTK, Bruton's tyrosine kinase. And this tyrosine kinase is an unusual tyrosine kinase, cytosolic, it's not transmembrane, and it has a plexure homology domain. And it was discovered because of its mutations in it. It resides on the X chromosome. It gives an X-linked agammaglobulinemia. In other words, B cells cannot activate, they cannot be stimulated by B cell receptor triggering if you have a defect in BTK. And what was striking is that the first mutation discovered in BTK to cause this was in the plexure homology domain in a crystal structure. And this actually led us, because we knew when we knocked out PI3 kinase in B cells that the B cell would not grow, very similar to agammaglobulinemia. So we asked whether the plexure homology domain of BTK might bind to PIP3, and it turns out that it did, as this crystal structure reveals, with the head group bound. And what's more, the arginine residue that's mutated to cause agammaglobulinemia is directly coordinating the 3-phosphate to PIP3, really solidifying the biochemical explanation of linking these two defects in B cell signaling through PIP3 and binding to the pH domain. And we showed that the wild-type BTK genetically prefers PIP3 over other phosphoinositides, and that mutation eliminates that preference for PIP3 binding. And this turns out to be a very high affinity interaction in the submicromolar KD. So other pH domains have also evolved to bind to PIP3. Not all pH domains do, but a subset do. And there are currently about 25 pH domain containing proteins that directly bind to PIP3. So we focus a lot on AKT being the major downstream signaling pathway, and it certainly is important. But keep in mind, it's not the only thing that PI3 kinase activates. There's a whole host of proteins with pH domains. So this little cartoon illustrates what happens when insulin or IGF-1 binds to the insulin receptor or IGF-1 receptor. First thing that happens is you get autophosphorylation of the receptor on tyrosine. That recruits IRS proteins to directly bind to the receptor, and they then themselves get tyrosine phosphorylated. And they are phosphorylated within a sequence motif that we showed back in the early 90s as optimal for binding to the SH2 domain of the regulatory subunit of PI3 kinase. And that motif is phosphotyrosine, methionine, X-methionine. IRS proteins have 7 to 9 of these. They're multiple IRS proteins. And as a consequence, they're professional gathering places for PI3 kinase. So with a single insulin receptor dimer, it's a covalent dimer, you can accumulate 2 IRS1 proteins, and each of them has 9 PI3 kinase binding sites, so you can actually bind 18 PI3 kinases to a single insulin receptor, and I only show one of them. But this then activates PI3 kinase and localizes it directly at the plasma membrane where the substrate PI45P2 resides. This allows PIP3 to be produced, and then these various pH domains come in to that location and get activated to do downstream responses. In the case of the serine-threonine kinase, AKT, it directly binds to PIP3. That partially activates, but what fully activates it is further phosphorylation by an upstream kinase called PDK1, and a second kinase, which I haven't shown here, called TORC2, which also has the ability to bind to PIP3. So that phosphorylates 2 different sites on AKT that fully activate it. Now AKT need not stay at the plasma membrane. It can drift off into other locations in the cell, even go into the nucleus, and then phosphorylate a host of different proteins that ultimately control the downstream response. At last count, I think there were about 150 substrates of AKT claimed in the literature, and I think probably at least a third of those are correct. It's always difficult to prove that a site on a protein is phosphorylated by a given kinase, and in many cases, sites can be phosphorylated by multiple kinases. But some that we have a lot of confidence in, because not only they have the right motif for being phosphorylated by AKT, but what's more, that motif is conserved through evolution back to flies and worms, include these proteins here. AS160, which controls glucose transporter movement to the plasma membrane. Tuberin, also called TSC2, which itself is a tumor suppressor. I'll talk about it in the next slide or two. That controls protein synthesis. Foxo transcription factors, which control genes involved in gluconeogenesis in the liver, but also can act as a tumor suppressor itself. GSK3, that, as the name implies, glycogen synthase kinase 3, controls glycogen in the liver and muscle, but also phosphorylates a host of different oncoproteins, including MYC, to control their degradation. So all of these proteins have been implicated in cancer to some extent, but they're also all implicated in controlling insulin signaling in the liver and in muscle. P10 then, of course, turns the system back off again, and as long as everything is working properly, you only turn on this growth signal when the cell needs to grow, and it gets turned back off once the cell has completed its task of growing. Now, if we look at what's downstream of PI3 kinase and just focus on AKT, it phosphorylates this protein, which I called tuber in the previous slide. The gene name is TSC2. It's the gene responsible for tuberous sclerosis, a hyperproliferative disease. And downstream of TSC2, we showed many years ago, is a RAS-like protein called Rheb, which is directly activating the TORC1 complex, which is a complex that includes a RAPTOR protein and MTOR. And this pathway, as I think everyone in the room knows, has been targeted for cancer with the rapalogs, rapamycin and everolimus and others. It has several downstream targets, but most interesting of which are enzymes involved in protein synthesis control, including S6 kinase and 4EBP1. And this ultimately can control the rate of protein synthesis, but also a host of other things, including lipid synthesis downstream of mTOR. Now, this pathway is regulated in a very complicated way. It senses amino acids. Work by David Sabatini at MIT has really elucidated a very interesting pathway involving another RAS-like protein called the RAG proteins that sense availability of amino acid in the lysosome. It's also regulated by AMP-dependent protein kinase downstream of LKB1, also called STK11. That is itself a tumor suppressor gene. And that happens in multiple ways, including AMP kinase phosphorylation of RAPTOR and also of TSC2. As a consequence, then, there's a very interesting mechanism by which this mTOR pathway is regulated. It's sensing the availability of amino acids, sensing whether or not ATP levels are sufficiently high, the ATP-AMP ratio, which is what regulates the activity of AMP kinase, but it also senses whether PI3 kinase is on through AKT phosphorylation of TSC2. And to further complicate things, TSC2 is also phosphorylated by ERK and by RISC downstream of MAP kinase. And as a consequence, it's redundant. There's redundant control mechanisms here, which extremely complicate therapies, because almost invariably, if you turn off the MAP kinase pathway, the PI3 kinase pathway gets hyperactivated. If you turn off the PI3 kinase pathway, the MAP kinase pathway gets hyperactivated. And RAS can activate both of them, although its major goal, RAS's major signaling goes through MAP kinase. It only tickles the PI3 kinase pathway. It does directly bind to PI3 kinase, but when you turn on RAS, you mainly get this pathway turned on, while with PI3 kinase, you mainly get this pathway turned on. Insulin receptor goes almost exclusively through PI3 kinase, while EGF receptor goes almost exclusively through RAS, even though they both can activate both RAS and PI3 kinase. So this is the complexity, and once you know this network, you realize the problems we have with targeted therapy. And I left off all the negative feedback loops that further complicate this. Just point out that everything in blue is a tumor suppressor, everything in red is an oncogene. And as you'll see in a moment, this network really controls cancer more so than any other network. One way of thinking about this is it's a computer AND gate. It's sensing, is there a growth factor? Is there essential amino acids present? Is there sufficient ATP? If so, then the network fires and the cell gets a signal to grow. If any one of these is missing, then the cell doesn't grow. But the mutations in that network allow the cell to think that it's having all these inputs even when it doesn't. And that's why you end up getting cancers with this mutations in this network. One very good example that really demonstrates quite clearly what's going on here is this a mosaicism disease, which was only figured out a few years ago to be due to mutations in PIK3CA. So these are not germline mutations, but rather mutations during development, an early stage in the embryo, where a single cell randomly picks up a mutation, a hotspot mutation, the very same mutations in PIK3CA that you see in head and neck cancers or other cancers, will spontaneously occur during development, and every cell of lineage downstream of that cell now has an advantage. It can take up glucose and grow at a much higher rate, and you end up with these really dramatically deformed limbs in which the only treatment is surgical treatment. PI3 kinase inhibitors are now going into clinical trials for patients that have these kinds of diseases. It can happen in any tissue, including in the brain. You get a macroencephaly in the brain. And there's also quite a bit of evidence that seizures, focal seizures, are often due to a mosaicism, hotspot mutations in PI3 kinase. So, as I say, everything in red is an oncogene, everything in blue is a tumor suppressor gene, and a lot of these things are targetable with drugs, and so there's a lot of interest in targeting it. Now, if we ask, how often do you get PIK3CA mutations in cancers? We've already talked about head and neck cancers here. If you add up hotspot mutations in green or amplifications in red, or the gray means that you have both an amplification and a hotspot mutation, then head and neck cancers add up to more than 35% have one or the other. They fall into company with other cancers that have these high rates, most of which are women's cancers, interestingly. Uterine cancer really is a champion, with more than 50% having PIK3CA mutations. Cervical cancers add about 50-50 mutations or amplifications. Breast, more mutations than amplifications. And ovarian, more amplifications than mutations. If you ask how often is anything in the signaling network, either receptor tyrosine kinases, RAS, PI3 kinase, mutated, in solid tumors, you can see that almost any tumor that significantly causes death in America is dominated by mutations in at least one node, if not multiple nodes in this network. Gray, again, means multiple nodes are mutated. So prostate cancer, for example, is mainly dominated by PTEN loss, but it's about 70% overall of some mutation in this network. So how do we deal with this? How can we use this information to get better therapies to patients? So if we just look at PI3 kinase inhibitors alone, I think at least 25 PI3 kinase inhibitors have entered clinical trials. Some have dropped out, but there are many still looking promising in clinical trials. And as I'll show you in a moment, it's pretty complicated. The one PI3 kinase inhibitor to be approved is adalimumab, and I'll tell you a little bit about that in a moment. There are also AKT inhibitors in clinical trials. There are attempts to activate AMP kinase for cancer treatment. And of course, there are rapalogs and catalytic site inhibitors of mTOR in trials. I left off all the things targeting the MAP kinase pathway, several of which have been approved, including RAF inhibitors. And of course, lots of tyrosine kinase inhibitors approved as well. So there turns out to be 4 different genes in humans that are capable of encoding enzymes that are capable of making PIP3, this triply phosphorylated lipid. They're called PIK3CA, B, D, and G. And 2 of them are primarily found in hematopoietic cells, PIK3CD and PIK3CG. But the other 2, PIK3CA and PIK3CB, are virtually in every cell in your body. So they're somewhat different in that the A form is activated only by tyrosine kinases, while the B form requires both the tyrosine kinase and a GPCR signal in order to be activated. So it's an integrator of GPCR and tyrosine kinase signaling. These 2, this one, the G form is specifically to GPCRs to activate it, and the delta form is very much like the alpha form, and it's activated only by tyrosine kinases. So there are drugs going into clinical trials for hitting all of these for various different reasons. These 2 over here are of a lot of interest with regard to immune events, and in case of the delta isoform, also for cancer. So this is what adalimumab targets, the delta isoform that turns out to be critical for B cell signaling. There are several inhibitors now that are specific for the alpha isoform, BYL719, the one that's been in trials the longest from Novartis, and I'll talk a bit about it. There are pan-PI3 kinase inhibitors, BKM120. I'll show some data on it, which is also a Novartis drug. Another drug that's looking very interesting in the clinic is GDC-0032 from Genentech. It's called the beta-sparing PI3 kinase inhibitor. It hits alpha, delta, and gamma, but not beta. And it has some selectivity, although not as much as we would hope, for the mutated form of alpha over the wild type. And this turns out to be important. I think if we could get inhibitors that were very selectively for the mutant forms, we would do much better in the clinic in the same way that the successful EGF receptor inhibitors are the ones that hit the mutant forms better than the wild type. It gives you a better window of efficacy toxicity. So the delta isoform turns out not to be mutated in B-cell lymphomas, but it turns out to be essential for the growth of B cells. As a consequence, we showed from knockouts many years ago, if you knock out, in fact, all the PI3, class 1A PI3 kinases in B cells, which is what we did in the late 90s, we impair virtually everything that's needed for a B cell to grow. You can't trigger the B cell to grow with anything. The same thing happens if you knock out BTK, which I showed you earlier directly binds to PIP3. So this network of B cell receptor activating PI3 kinase, which then produces PIP3 to activate both BTK and AKT, is really driving everything that the B cell does. And as a consequence, both PI3 kinase inhibitors, the delta-specific inhibitor, as well as the BTK inhibitor, went into clinical trials for treating B cell-derived lymphomas. This is the kind of waterfall plot that everybody wants to see in a clinical trial, where virtually every single patient that has CLL, chronic lymphocytic leukemia, which is a B-cell lymphoma, responds. Indolent non-Hodgkin's lymphoma in general, mantle cell lymphoma, some pretty dramatic waterfall plots. And this kind of data led to accelerated approval, and adalimumab was approved last July. This is an example of a woman who was a patient at Weill Cornell. She had a B-cell lymphoma. The swelling in the neck is characteristic of this. She was only 38 years old, but she'd already failed all therapies and standard of care for CLL and was seeking clinical trials. She went on the adalimumab, and this is what she looked like 3 months later. So that swelling goes away very dramatically, and she was stable for at least 3 or 4 years before she ultimately progressed in this disease. So it was this kind of response that got the drug approved. So how do we extend beyond this? The advantage of targeting the delta form of PI3 kinase is there are very few side effects as a consequence of hitting that isoform. It's not mediating insulin signaling. It's not in the insulin signaling pathway. Solid tumors don't even, or normal tissues don't even have this other than the lymphocytes. So there's a nice window of efficacy toxicity, and the one side effect, the major side effect of this is a change in a suppression of Tregs, so that you actually get some autoimmune like colitis, which goes away when you take the drug away. So what about targeting PI3 kinase in solid tumors? So I was fortunate enough to get a $16.5 million grant from Stand Up to Cancer, AACR, several years ago to put a team together to try to figure out how to use PI3 kinase inhibitors in drug combinations to treat women's cancers. And the reason we focused on women's cancers is because of those high rates of mutations that I showed you earlier. So the team included Gordon Mills, my co-leader. Charles Sawyers initially was part of our team, but ultimately went on to start a second team with focusing on prostate cancer. Tom Robertson, Eric Weiner from Dana-Farber were heading that site. Ramon Parsons at Columbia, David Solit took over Charles's site as leader at Sloan Kettering, Carlos Artiega at Vanderbilt, and Jose Baselga. Then at Val d'Hebron, when we started the team, he moved to Mass General. We brought in Mass General. He then moved to Sloan Kettering. In fact, he and I moved to New York together as a team and started a journal together, Cancer Discovery. So it was clear from the very early work, particularly work being done in Jose Baselga's group at Val d'Hebron, that these waterfall plots are not as impressive as the plots that we see with idelalisib. So this is a trial in which every patient in this trial has a PIK3CA mutation. So we had a question to Peter earlier about this. Is there a correlation between having a mutation and response? And clearly a lot of patients with mutations fail to respond. Now the different colors here indicate different solid tumors, so that the pink is breast and this yellow-orange type color here is colorectal cancers. And it's clear there's a lot more pink over here and a lot more orange over here. We can explain some of these failures to respond by the fact that these colorectal tumors often have a KRAS mutation and a PI3 kinase mutation in the same tumor. Now, at the time that this was done, figuring out whether these were truncal mutations or branch mutations, the technology wasn't sufficiently good. So it could also be that some of these are really 1 or 2 or 10% mutated in the tumor. So this needs to be reevaluated. But I think there are other reasons than just whether it's a truncal mutation or not that's explaining why the patients don't all respond. So we need something better than just do you have a point mutation to decide who's likely to respond. So I mentioned earlier that the major goal of PI3 kinase is to drive glucose uptake in the cells. You activate PI3 kinase, it drives glucose transporters to the surface of the cell and drives multiple steps in glycolysis. And the first observation that glucose was taken up at a high rate in cancers was made by Otto Warburg more than 90 years ago, in which he observed that a rat tumor induced by mutagenesis the tumor took up glucose 50 to 100-fold faster than the normal tissue from which it evolved. And this, of course, led ultimately to technologies of FDG PET as a way to identify cancers. Now, I should say that all cancers don't have dramatic glucose uptake, but most do. Also, we now know that all cancers don't necessarily use PI3 kinase to drive glucose uptake. There are other ways to do it as well. MYC-induced GLUT1 expression, for example, is another way for this to happen. But we ask in a mouse model, if we drive a cancer with PIK3CA mutation, in this case this was a lung adenocarcinoma that Jeff Engelman and Kwok Wong worked on when Jeff was a postdoc in my lab. And if you express this PIK3CA mutant tumor, this is putting human PIK3CH 1047R mutation into the lung. After about 12 weeks of expressing that gene, you get a lung adenocarcinoma. And this is screaming hot by FDG PET. If we now turn off PI3 kinase by taking the doxycycline, which is needed to drive the expression of the human gene in the mouse, then that goes away. Or if you give a PI3 kinase inhibitor, BZ235, that glucose uptake goes away. The tumor's still there. You see this response within 48 hours, but the tumor's still there. It just doesn't take up glucose. And ultimately, these tumors shrink. On the other hand, you have a KRAS-driven lung adenocarcinoma. The pathology is it looks identical. You give it the exact same drug, and nothing happens at all to the glucose uptake. So the RAS-driven tumor doesn't use PI3 kinase to drive glucose uptake. It's doing it by another mechanism, probably through MYC expression. And as a consequence, the PI3 kinase inhibitor does nothing to affect the growth of this tumor. If you can get enough PI3 kinase inhibitor and MEK inhibitor simultaneously in the mouse without killing it, you can make these tumors go away. But that's a problem we have in human clinical trials. It's clearly the combination you'd like to use, but the toxicities are really quite difficult to manage. So this raised the question, can we use this kind of functional assay to determine who's likely to respond to a PI3 kinase inhibitor? So I would predict that if you can't turn off the PET signal with a PI3 kinase inhibitor, you're not going to benefit the patient. So we asked that in the clinic. This was an example of a patient who did have a nice PET response with after 1 month of therapy, where you can see this liver met, the liver met FDG signal completely goes away. And the tumor's still there, I should say, after a month. It hasn't disappeared, but it clearly can't take up glucose anymore. And ultimately, this patient had a pretty dramatic response to this and benefited for more than a year with tumor shrinkage before a new metastasis appeared. By the way, this patient did have a PIK3CA mutation, ER positive, HER2 positive, PR positive, went on all the standard of care that you'd expect, Lupron, Herceptin, Lapatinib, progressed and all that, and then went on to a single agent PI3 kinase inhibitor and still had this dramatic response. So this was probably a truncal mutation. So we asked, we didn't do enough patients to try to prove statistically that it was true that you could predict who's going to respond by repeat FDG PET. And I should say I wanted to do the repeat PET within 48 hours because we know that works in the mouse. The doctors explained to me that it's not very convenient for the patient or them to do 2 PETs within 48 hours, so we decided on 2 weeks. And so this waterfall plot is not the size of the tumor, but the area under the curve for the PET signal. And so these patients over here had a decline in FDG PET following 2 weeks of taking PI3 kinase. These 2 patients had actually an increase in FDG PET 2 weeks later. And then we had some that had very little change. And the color here indicates, the blue indicates that these are the patients who had a clinical response. And it's at least a 30% shrinkage in tumor. And this patient actually had progressive disease, and these had stable disease. So there's a nice correlation in this small population of patients that did this. It predicts maybe this would be valuable in the clinic. We published this result last year in Journal of Clinical Oncology. And listed here. I won't go into all the details of this. You can go read the paper. But the bottom line was that we had a lot of patients who did not have PIK3CA mutations who still responded to single agent PI3 kinase inhibitor. And we've done this both with BKM120 and also BYL719. And based on these results, Novartis has decided to do a neoadjuvant trial. This was really a lead-in to a phase 2 neoadjuvant trial of ER-positive breast cancer patients divided into the subset that have PIK3CA mutations, which is about 35 to 40%. But we also have an arm of patients who don't have the mutation. And then we'll look for pathological responses in those patients. And that trial is now enrolling. It will enroll at least 600 patients worldwide. Now, we also decided to look into triple-negative breast cancer, and triple-negative breast cancer is really an outlier, as the name implies. It's a cancer we don't have any therapies for, or at least any targeted therapies for. It's ER negative, PR positive, PR negative, and HER2 negative, and it's now the disease that has the worst outcome. Does respond to chemotherapy, but almost invariably comes back and metastasizes. These tumors have only about 8% of them have PIK3CA mutations, but the majority of them have PTEN loss and another phosphatase IMPP4B loss. Almost all of them, or a large fraction of them, have p53 mutations. And in fact, mutationally, it looks more like ovarian cancer than the other 2 subsets of breast cancer. And in fact, I think as everyone in the room knows, these are also the 2 diseases that frequently occur in BRCA patients. And so we imagine in the evolution of ovarian cancer and triple-negative breast cancer is really taking a very similar course, and are beginning to try to understand this at the molecular level by sequencing those tumors. So we made a mouse model that was BRCA deleted and p53 het. This is germline p53 het, breast-specific BRCA deletion, and tested the PI3 kinase inhibitor. Now, these tumors take about a year to appear, but once they appear, it only takes about a week for the tumor to get to a size where they force us to sacrifice the mouse. Because these tumors divide about every 16 hours. They double in size every 16 hours, so very rapidly growing tumor. And so you can see our control group, they all have to be sacrificed in about a week. The PI3 kinase inhibitors actually had a pretty good effect. BZ235 extended out to a couple of weeks, BKM120, we can get almost a month out. But no mouse was cured with a single agent PI3 kinase inhibitor. We tried BYL719 as well. So if this were a human trial and you saw that much life extension, you'd probably approve it, particularly if you called this months instead of days. But no home runs. But we noticed as we characterized these tumors as they became resistant, we've now sequenced these tumors, the primary tumor and then the tumors as they become resistant. And we have now over 100 tumors sequenced, both full exome sequencing and RNA-seq, to try to determine the mechanism of resistance, and also the evolution of the primary tumor. takes a year for it to evolve. What happened during that year that made the tumor take off? And very interesting results are coming out of that. But in the course of characterizing them, we realized that when we added a PI3 kinase inhibitor, we got an increase in DNA damage. And with BRCA loss and p53 loss already, you have a fairly high level of DNA damage as judged by gamma H2X and poly-ADP ribose. But you can see this is dramatically enhanced when you add the PI3 kinase inhibitor. This is actually human cell lines, but we saw the same thing in the tumor. So only in the context of having BRCA and p53 mutations do you see this kind of massive DNA damage. So, of course, PARP inhibitors were already in clinical trials at the time we made this observation a little over 3 years ago, so we decided to test PARP inhibitors in this setting. And the one drug that was available at that time that was most furthest along in clinical trials was olaparib. Olaparib was, in fact, now approved, but at the time we did this study, it was not. So we tried olaparib, and it works. You know, you can see almost every tumor responds to olaparib, but it's no home run either. They all become resistant within less than a month. So not quite as good in this mouse model as the PI3 kinase inhibitor, but it certainly had an effect. So we then asked what happens if we put the two together, and there we saw a much more dramatic effect. We're now out, 3 months or so, although most of these tumors ultimately went on to progress. So we had another model in which both p53 and BRCA were fluxed, and we asked what happens in this model. So now it only takes 7 months instead of a year for a tumor to emerge. And this is now a log plot, I should say, rather than a linear plot, so the curves look somewhat different. But again, once these tumors appear, we have to sacrifice the mouse within a week or so. Now, BKM 120 was not as dramatic in this. Certainly most mice responded to this drug, but not a very durable response. And the alpha-specific PI3 kinase inhibitor really had virtually no effect on these tumors compared to the control. But olaparib actually works better than PI3 kinase inhibitors in this setting. But again, no cures. We got what you would call stable disease, but ultimately they took off again. But when we put the two together, every single tumor responded and completely went away. There was no tumor left at all. And we took these out now for 6 months, and some of them, in fact, for a year and a half, and the tumor never came back. So I should qualify this by saying that we transplanted these tumors into littermates. This is a highly inbred model, and so you can transplant the tumor into cleared fat pad of a littermate so that you don't get any new tumors forming. Because otherwise, if you do it in the original mouse, every single cell in every breast has p53 and BRCA, and so you'll continually over the life of the mouse get new tumors appearing. But here, once you transplant the tumor, you can actually cure it. This is what the Kaplan-Meier curve looks like. Single agent, virtually, that's, you would say, why would I even bother to combine these with something? There's nothing, no response there, hardly any response, particularly with BYL719. But BYL719 is actually, when combined with olaparib, is the best combination we've yet to find. BKM120, very similar. And so that stimulated us to do a phase 1B trial of combining these two drugs. We called up Novartis because I'd signed an MTA saying I would never combine their drug with any other drug. And I explained how the technician had accidentally put the two drugs together. And they said, find us some company that will work with us, and we'll do a trial. And so I called up AstraZeneca. They agreed to meet at ASCO with our clinical team. We showed them the results, and they agreed to give the drugs to us for the trial if I would pay for the trial. But I had, still about $10 million left from the grant, so we decided to go forward with it. And I give Ursula Matsalanis a lot of credit for moving this through the SRC, the IRB, and the FDA to get this in record time. This trial started 3 years ago, almost in September 2012. And the data was presented at the AACR meeting, at the annual meeting in April. And here's, I'll show you some of the data from the trial. So we decided to enroll both breast cancer patients and ovarian cancer patients, given what I told you earlier with the very similar mutational background, even though our preclinical data was only in breast. We had more ovarian cancer patients than breast. This opened in multiple centers. In fact, almost all the centers of our team ended up enrolling patients in this trial. And you can see some nice waterfall plots here. And what's even more importantly, there were durable responses. So here's the number of responses seen. None of the patients actually got a complete response as judged by complete disappearance of tumor, but partial responses defined by at least a 35% reduction in tumor. We had quite a few of those. About a quarter of the patients had a partial response. Lots of stable disease. So between stable disease and partial responses, about 75% of the women, both breast and ovarian cancer, showed that kind of response. And what's more important, the second patient to ever go on the trial, an ovarian cancer patient at Beth Israel Deaconess, one of Gerber-Wolf's patients, is still on the trial today, 3 years later, which I'm told, I'm not an ovarian cancer oncologist, but all these patients had progressive disease and had failed all standard of care, including platinum treatment, at the time they went on. So this seems to be a pretty remarkable response. So everything with an arrow here indicates those patients are still on the trial. We've quit enrolling in the trial because we've reached, our phase 1b dose for the patients, for the phase 2 dose for the patients. But all these with arrows are still on the trial, for several for over 2 years. So here's just an example of a patient who had a liver met, it's a lung met rather, and almost a complete response where this metastasis goes away. This was a triple-negative breast cancer patient, and here's a patient, ovarian cancer patient, that had a quite significant and durable response. So conclusion then is that figuring out how to use PI3 kinase inhibitors in solid tumors is going to be complicated. We almost certainly have to use them in drug combinations, and figuring out what the right combinations are going to depend on having some really reliable preclinical models, PDX models, as well as genetically engineered mouse models, to figure out what's tolerable. Can you get the right dose, get an efficacy toxicity ratio that looks impressive enough to go into a human phase 1b trial. The delta inhibitor was already approved. The advantage there is you could get quite high doses because the side effects were pretty minimal, mainly autoimmune responses, which were an on-target effect. The problem with the alpha-specific inhibitors and the pan inhibitors is they have the on-target effect of turning off insulin signaling. And so the patients, as soon as they go on a dose that's going to be effective in hitting the PI3 kinase in the tumor, are going to get hyperglycemia. I remember when we first started talking to companies about our team and how we would like to try their drugs, many of the pharmaceutical companies would come to me and say, use our drug, it doesn't cause insulin resistance. And I said, well, if it doesn't cause insulin resistance, it's not hitting PI3 kinase, why would we use it? And so that's, if you don't see insulin resistance, you know you're not hitting the target hard enough. You should see dramatic insulin resistance. And then the question is, how do you handle that? So for our Stand Up to Cancer team, I tell everyone, first of all, the patient's got to go on a ketogenic diet. You've got to keep glucose levels as low as possible when taking these drugs. And then secondly, you need metformin. You never want to give them insulin. If you give them insulin, it will activate PI3 kinase in the tumor again, because most of these tumors have high insulin receptor. And it's probably insulin that's driving the tumor. And the reason patients who don't have a mutation are responding to the drug is because it's insulin that's driving the PI3 kinase, not the mutation. So figuring out how to use these drugs keeps the patients off sugar, off any carbohydrates as possible, give them a ketogenic diet, put them on metformin. There's also a sodium glucose cotransporter inhibitor just approved that you can also lower glucose levels. And that's, I think, if you can't manage the patient on those combinations, then you should get them off the trial. Obviously, it could be a home run if someone could make inhibitors that only hit the mutant PI3 kinase and there's certainly a lot of interest in doing that, but we're not really there yet. So I think I'll finish with that and just, again, acknowledge the team, the Stanford Cancer team that worked together on this, and particularly Gerberg Wolf's lab, where I'm still collaborating. So thank you very much.
Well, thank you very much for a fascinating talk. So we have a few minutes for questions. Are there any questions from the audience? And please use the stand-up mics. So I have a question. So I probably should go and use the mic as an example. So when you look at the effect of these inhibitors, you focus a lot on the size of the tumor, but did you ever look at what happens to some of these cancer-initiating cells or some of the pathways that have been associated with initiating? What happens in response to these inhibitors?
What happens to the pathways?
Right. I mean, are you activating like Wnt pathways? Did you look at some of the other maybe pathways that are more associated with primitive cell state?
Yeah. No. I mean, the problem is for these phase 1 trials, it's really hard to get biopsies, mandatory, but nobody wants to do mandatory biopsies in the setting of the trial. And so even though I really encouraged all the clinicians to get biopsies before, acutely on the metastatic disease and after therapy, or particularly after they've become resistant, we had very little success in that. Now, as we're going into phase 2 trials, and particularly the neoadjuvant trial, we'll have that data because you do a biopsy to diagnose the patient, so you know what's going on in that acute biopsy, and then there's surgery at the end, so you know what's going on after the therapy. But right now, we really have a limited number of patients where we have that access to the fresh tissue. Of course, in our mice, we do have that, and we are looking at every possible thing in the mouse, including, as I said, RNA-seq before and after treatment, RNA-seq before and after resistance emerges, and that's really telling us a lot. It's going to take forever to analyze all the data coming in from that, but we're already seeing things that begin to make sense. I should say that as these cells become resistant to PI3 kinase inhibitors, the MAP kinase pathway comes up in these mice. So as they become resistant, we don't see elevated AKT, so they're not going around the drug by reactivating PI3 kinase, but they're hyperactivating the MAP kinase pathway. And we're beginning to understand how they're doing it. It's usually FGF receptor. And so that's, sometimes it's MET, tyrosine kinase. So we're getting tyrosine kinases turned on by fusion events and by amplification events that are giving us resistance.
Okay, thank you. So you partially answered my question of what other pathways will be activated in the resistance. But my next question is the resistance tumor, do they still have PIK3CA mutation or amplification, or it is a different part of the heterogeneity of the tumor that they don't have it?
Yeah, so the tumors, you mean the tumors that we engineered into the mice, or the tumors from the human?
Tumors in the patients.
In the patients. Yeah, so these, we have done analysis of the patients on this trial I just described. We have mutational analysis on 40 patients from that trial. And they have the things you'd expect to see. Some of them have PIK3CA mutations, some have P10 loss, a lot of them have P53 mutations, the majority of them have those. We tried to correlate these durable responses with a potential mutational signature, and nothing really emerged. We can't, I think the number of patients was just too small for a pattern to appear there.
Do you think that germline mutations that these patients have may be contributing in the resistance?
Well, so we certainly analyzed BRCA germline in all the patients. About 50% of the patients did turn out to be BRCA germline, both in the triple negative subgroup as well as in the ovarian subgroup. That seems relatively high nationwide that you'd find that percentage, but in fact, most of our patients came from New York or from Boston, Dana-Farber, Beth Israel Deaconess. So we had certainly a disproportionate fraction of women, Jewish women who were in these studies, and that accounts for it. But being BRCA mutant was not necessarily correlating. It slightly correlated, but it didn't. There were lots of non-BRCA patients who responded. So a phase 2 trial is going to start probably beginning of the year with BYL719 plus olaparib. And again, we will include BRCA mutant and BRCA wild type as well. The trial isn't fully designed yet, but that's the plan.
Thank you.
I really enjoyed your talk. I had a question. For these trials, given that lymphocytes express the delta and gamma subunits, whether you guys are looking at the impact of pan class 1 inhibitors like BKM120 versus the alpha-specific inhibitors, whether that you're seeing differences in immune function impact or not.
Yeah. So that's a really important point. As I say, what we did like 10 years ago is do a T-cell specific deletion of all class 1A PI3 kinase. So we didn't knock out the gamma form, but we knocked out alpha, beta, and delta. And so all the tyrosine kinase signaling group we knocked out. And the phenotype was quite disappointing to the student. It was actually a collaboration with David Fruman's lab, because he couldn't see a phenotype. And it shocked me that, given that I published numerous papers on T-cell using PI3 kinase to trigger virtually everything, calcium responses, et cetera. But it turns out that in vivo, you don't need PI3 kinase, at least class 1A PI3 kinase, in the T-cell in order to get a response. And as a consequence, we had no defects in immune response with knocking out all the class 1A PI3 kinase. So the dendritic cells can still present antigen and activate the T cell without the need for PI3 kinase. But we noticed the mice were scratching their eyes, and it turns out they developed autoimmune disease. So the one T cell population that needs PI3 kinase is the Tregs. So these pan PI3 kinase inhibitors, and as well as even the delta inhibitor, as I said earlier, give you an autoimmune disease. And that's not because of the B cell effect, that's because of the T cell effect. And the Tregs are getting suppressed in the human case as well. So one way to consider PI3 kinase, and it's even possible that some of the responders are responding because we've activated the immune system by suppressing Tregs globally in the body. And so that needs to be looked at more carefully.
Okay, well, thank you very much, and we will then proceed.